Hello guys, ngayon di-discuss natin ang DNA as the genetic material. So dito, matatakal natin, review lang dun sa pinanood ko sa inyo na video, short review lang dun sa experiments, and then DNA structure, and then DNA as the genetic material. DNA as genetic material, it was first extracted from nuclei in 1870, named nuclein after their source. Ang chemical analysis determined that DNA was a weak acid rich in phosphorus. So please take note of this. Ayan. Rich in phosphorus. It is a weak acid rich in phosphorus. So its name provides a lot of information about DNA. Deoxyribose nucleic acid. Okay. Contains a sugar moiety. Yung deoxyribose, it is weakly acidic and it is found in the nucleus. So, deoxyribose nucleic acid. Because of its nuclear localization, subsequent identification as a component of chromosomes, it was implicated as a carrier of genetic information. Are genes composed of DNA or protein? Chromosomes are also known to contain protein, pero ang chromosomes kasi natin, sila ay bundle of DNA. So, tightly packed DNA. So early on, it was a challenge to demonstrate that DNA was indeed the molecule that contained the genetic information. DNA, only 4 different subunits make up the DNA. Chromosomes contain less DNA than protein by weight. Weight lang ang basehan nito. Kasi actually, sobrang bigat ng protein kasi sa mga biomolecules natin. By weight, medyo mas mabigat talaga ang protein kaysa sa DNA. Ang protein kasi may 20 different subunits. Ang DNA natin, 4 lang. Greater potential variety of combinations. Chromosomes contain more protein than DNA by weight. Classical experimental data confirm that DNA is the genetic material. So, bacterial transformation implicates DNA as the substance of genes. Ito, yung experiment ni Frederick Griffith noong 1928. Gumamit siya ng smooth. Yun yung virulent strain ng Streptococcus pneumoniae and rough, non-virulent strain. Si Oswald Avery, Colin MacLeod, and Maclean McCarthy determined that DNA is the transformation material. So, ito yung kay Griffith. Babalikan natin yung picture mamaya. Observed that the live S bacteria could kill mice. Live S bacteria could kill mice injected with them. When he hit-killed the S variants, hit-killed na, and mixed them with the live R variants, then injected the mixture in the mice, they died. Pero nung hinit-kill lang yung S variants, tapos in-inject na dun sa mice, hindi namatay kasi hit-killed na yung variants. So, Griffith was able to isolate the bacteria from the dead mice and found them to be of the S variety. Thus, bakit ganun? Pinatay na niya yung S variants and then hinalo niya yung R variant na hindi naman dapat fatal dun sa mice. Hindi naman yun yung virulent strain. Thus, the bacteria had been transformed from the rough to the smooth version. Kasi in-isolate niya, nag-isolate siya ng bacteria dun sa namatay na mice, dun sa may heat-killed na S, and then nakamix na R, live S ang nakuha niya. The ability of a substance to change the genetic characteristics of an organism is known as transformation. Scientists set out to isolate this transforming principle since they were convinced it was a carrier of the genetic information. So, andito siya. R strain, no components destroyed. R strain, mouse dies. Polysaccharides destroyed. Ito dead. So, hit killed. Ang pangit ng sulat ko. Hit killed S. Hit killed S. So, minix niya dun sa R strain, namatay. And then, polysaccharides destroyed, namatay si mouse. So, ano meron kung polysaccharides? Ito kasi, yan na yung sugars. Tinanggal nila yung possibility na sa carbohydrates mo kuha yung genetic material. Lipids, tinanggal nila yung possibility na sa fats mo kuha ang genetic material. So, ito RNA, alam nyo na yan. Tinanggal nila yung possibility na sa RNA manggagaling ang genetic material. Tinanggal din nila ang protein, namatay siya. And then, pinatay ang DNA, nabuhay si mouse. Ibig sabihin, no life strain recovered. Nabuhay siya nung DNA ang dinestroy. Okay? Hershey and Chase experiments. In 1952, si Alfred Hershey and Martha Chase provided convincing evidence that DNA is the genetic material. Waring blender experiment using T2 bacteriophage and bacteria. Radioactive labels, ito si phosphorus 32 and sulfur 35 for protein. Ito. Kasi, remember, ang DNA rich in phosphorus. And ang proteins natin rich in sulfur. Ayan. So, yun yung tags na ginamit nila. Ayan. So, dito, sa una, ito yung first, ang naka-radioactive label, yung mismong bacteriophage, yung katawan niya. Yun yung red. Yun yung may radioactive label. So, sinubject siya sa bacteria, which is E. coli in this case. And then, na-detect nila yung radioactive label dun sa katawan mismo nung T2 phage, walang nangyari. So, andun lang siya. And then, sa pangalawa naman, yung may radioactive label, yung DNA. So, ang T2 bacteriophage kasi natin, protein and DNA lang laman. So, maganda talaga siyang subject ng treatment kung subject ng experiment kung DNA nga ba, ang DNA or protein nga ba ang source ng genetic material. So, itong sa second, yung DNA, ang may radioactive label, yung 32P. So, na-subject ulit siya sa, yung E. coli, na-subject ulit sa T2 phage. And then, nakita nila, same treatment, na blender and na centrifuge, na-detect nila yung radioactive label sa loob ng bacteria. Tapos, nag-proliferate siya. Ibig sabihin, nag-multiply siya. Dumami yung number niya, nung radioactive label. The phage are made up of The phage are made up of equal parts of protein and DNA. It was known that the phage infected by anchoring the outer cell to the cell surface and then deposited to the inner components of the cell, infecting it. Scientists were interested in finding out whether it was the protein component or the DNA component that got deposited inside the infected cell. By incorporating radiolabel either in the protein or in the DNA of the infecting phage, they determined that the DNA was indeed introduced into the infected bacteria, causing proliferation of new phage. So, si Watson and Crick naman, DNA sa double helix. Si James Watson and anong pangalan ni Crick? Nakalimutan ko. Knowledge of the chemical structure of nucleotides, ito deoxyribose, phosphate, and nitrogenous base, ito yung main components ng DNA natin. Si Erwin Chargaff, siya ang nag-determine na ang ratio ng adenine sa thymine are always 1:1, ang ratio ng guanine sa cytosine are always 1:1. Kasi ganyan lagi ang pairing. Pairing ay A, T, C, G. Always yan, hindi magpapalit. Hindi magpe-pair si C at T, hindi magpe-pair si A at G. Always, A kay T, C kay G. In 1953, James Watson and Francis Crick proposed their double helix model of DNA structure. Ang issue dito actually, hindi nila binibigyan ng credit si Rosalind Franklin. Ang study ng double helix structure nagsimula kay Rosalind Franklin. Yun ang issue ng mga biologists ngayon. Hindi nila binigyan ng credit si Rosalind Franklin. Nakakuha sila ng Nobel Peace Prize kung hindi ako nagkakamali. Okay? Sa X-ray diffraction patterns produced by DNA fibers, si Rosalind Franklin and Maurice Wilkins ang kumuha ng image na to. So sila ang nagsabi kina Watson and Crick na, nakakuha ko ng X-ray pattern, feeling ko double helix structure ang DNA. Personally, hindi ko nakikita. Paano kaya nakita ni Rosalind Franklin yon? Hindi ko alam. Pero siya ang nagsabi kina Watson and Crick na maaari, siya ang nag-propose na maaari ganito ang structure ng DNA. Si Watson and Crick, sila ang nagpatunay. Pero nagsimula lahat kay Rosalind Franklin at sa picture na to. Dito sa X-ray diffraction pattern na to. Okay. So, DNA's chemical constituents. Number 1 is deoxyribose sugar. So, may pentose sugar. Pentose kasi five-sided. Five-sided na ayun. Basta ganun. May deoxyribose sugar siya. Pangalawa, always na may phosphate group. Una, may deoxyribose, kaya nga deoxyribose nucleic acid. Pangalawa, always siya na may phosphate group, kaya ang DNA natin rich in phosphorus. And thirdly, one of these 4 nitrogenous bases. So, sa nitrogenous bases, may dalawang klase. May purines, may pyrimidines. Ang purines generally bigger kasi may 2-ring structure siya. Tapos ang pyrimidines natin, smaller. Iisa lang yung ring structure niya. Si adenine at si guanine, ang purines natin, and then sa pyrimidines, si thymine at si cytosine. Ayan. Ganyan sila pag magkakasama. Pag si deoxyribose lang and si nucleotide lang ang magkasama, ang tawag dun, nucleoside. Ito, pag kumpleto, meron siyang phosphorus, meron siyang deoxyribose, meron siyang nucleotide. Ang tawag dyan, bakit ganun? Ayan, mali ako, sorry. Balik tayo. Pag deoxyribose lang, plus isang nitrogenous base, ang tawag dun, nucleoside. Pero, kung dinagdagan mo ng phosphorus, kasama niya si deoxyribose and then nitrogenous base, ang tawag doon, nucleotide. Ito, since dalawa ang ring structure niya, purine siya. And then, dito naman, since isa lang ang ring structure niya, pyrimidine nucleotide. Ang purine natin, kasama dyan, ay ang adenine and guanine, AG. And then sa pyrimidine, meron si C at si T. Sorry, ayan. Ito, meron akong nilagay na sample computation. Try natin. Di ba sinabi, ang pairing ATCG, always yan. So, laging equal ang bilang ni adenine kay thymine, laging equal ang bilang ni cytosine kay guanine. So, supposedly, for example, sa isang DNA strand, 30% ay adenine. 30% ay A. Ilang percent naman? Ito muna ang tinitignan natin ha. Pag 30% ay adenine, ilang percent si thymine? Ang sagot, 30% din. Pero paano pag ang tanong, 30% si adenine, ilang percent naman si guanine? refer tayo dito sa blank space dito sa baba. So, ang given ay 30% A. Ilan ngayon si C? Ilang percent? Since 30% si A, for sure, 30% si T. Kasi lagi silang equal. Equals 60%. Pinag-add ko lang. So, 100 minus 60 is equal to 40. Since laging equal si C kay G, hati sila ngayon sa 40%. So, 40% divided by 2 is equal to 20%. Yan ang sagot. 20%. C is equal to 20%. Madali lang. Kasi, given, kahit isang nitrogenous base lang ibigay sa'yo, makukuha mo yung percentage ng lahat. Okay? DNA's chemical constituents part 2, nucleotides linked in a directional chain. So, ayan. Malalaman mo ang direction ng DNA. DNA is polar 5' to 3'. Ito, 5' end. Tama ba? Ito, 5' end, 3' end. Iisa lang yan. Sabihin na natin na dyan si number 1, meron number 2 dito. And then, lagi silang magkabaliktad. Since 5' end ang nandito sa taas, 3' end ang nandito. Andito naman sa baba niyan, 5' end. Ayan. Magkamukha lang yan. Beta and glycosidic bond connects the sugar to the nitrogenous base. Yung sugar natin, yun yung deoxyribose. A phosphodiester bond connects one nucleotide to the next. Ang nucleotide, yan ay kumpletong phosphorus, deoxyribose, and nitrogenous base. Huwag niyong kakalimutan yon. So, di ba sabi ko, ito kunwari, isang nucleotide to, merong nagko-connect dito na bond. So, ito yung nire-refer niya. Kasi, itong nakabox, isang nucleotide, ito, isang nucleotide, merong phosphodiester bond na nagko-connect dyan. Okay? The sugar phosphate backbone is identical in every DNA molecule. In any DNA, purine is equal to pyrimidine. Watson and Crick model, double helical structure daw siya. The two strands are anti-parallel. Kaya ang sinabi ko kanina, di ba? Ganito ang DNA natin, double helical structure. Pero imagine niyo pinagdiretso natin siya. So, ganyan. Straight na siya. Imagine niyo na lang ha, in-unwind natin yung DNA. May 5' end ka dito. May 3' end sa baba. 5' end dito. 3' end. Kaya anti-parallel. It is a right-handed helix, so ang DNA natin laging paikot toward the right. Ganun ang laging ikot ng DNA natin. This structure is called the bDNA. Complementary base pairing, 3 hydrogen bonds between C and G. Ganun ang pairing ng C and G. 3 hydrogen bonds. So, kada line hydrogen bond yan. 2 hydrogen bonds between A and T. Ayan. Hindi nyo makikitang magpe-pair si A at G. Hindi nyo makikitang magpe-pair si C at T. Never mangyayari yon. Laging CG and AT. And sinasabi dito, 3 hydrogen bonds kay cytosine and kay guanine, 2 hydrogen bonds kay adenine and thymine. The arrangement of the nitrogen bases determine the genetic message. At each position, there are 4 possibilities. Therefore, for 100 base pair long molecule of DNA, there are 4 raised to 100 variations possible. Okay. So, ito, laging 4, and then, aalamin mo na lang kung ilang base pair. So, ganun lang yun. Madali lang yan. Complementary base pairing, ayan, ganyan siya. So, si C, ay si G, si C, A, T. Chargaff's rules. Ratios of nucleotides, A is equal to T, and G is equal to C. A plus T does not have to be equal to G plus C. Ayan. Para hindi ko na kayo lilituhin, mag-example na lang tayo agad. Later. The structure of DNA, part 2. Ito yung major groove. Ito. Nakikita nyo naman sa Nakikita nyo naman sa illustration. Pero ito. Basta yung malaking part. Yan. Yan yung major groove. Minor groove, major groove. Magigets yun na yan. Ito yung base pairs. Andyan yung nucleotides natin. Magka-connect na sila. Ito yung sugar phosphate backbone. Yung blue. Yung base pairs natin, yung yellow. Yung major groove, yung image na nakikita nyo, ayan. Kung ano yung mas mahaba, yun yung major groove. Kasi, lagi mo lang siya makikita sa isang side. So, kung ano yung mas malaking portion, yun yung major groove. Tapos, yung paikot na part, yun yung minor groove. Kasi, mas maliit na siya eh. Question. DNA was extracted from cells of Staphylococcus and found to have 37% cytosine. What percent of guanine does this species have? Ayan. So, ang given C ay equal to 37%. Ang hinahanap ay si G. Si C equal kay G. Madali lang to. Ibig sabihin si guanine ay equal to 37%. 'Di ba? Madali lang. Pero baguhin natin yung question. I-erasing ko to lahat. Ulitin natin. So, ang sagot ay 1. Pero, ulitin natin yung question. Tatanggalin ko to. Okay. So, let's change the question. Ay, sorry. Wag naman guanine. Paano pag ang tinanong dito ay thymine? What percent of thymine does this species have? Okay. Ang given, cytosine is equal to 37%. Ayan. Ang cytosine natin, 37%. Ang hinahanap natin ngayon, si thymine. So, si C equal to G, ibig sabihin si G is equal to 37%. So, 37 plus 37 is equal to 74%. Yan na si C and G. So, ilan na lang natira? 100 minus 74 is equal to 26%. Si 26% is equal to thymine and adenine. Sila na lang natira eh. So, si thymine ngayon, i-divide mo lang sa 2, yung 26, is equal to 13%. Ganun lang kadali. Okay? I hope naintindihan ng lahat. What percent of thymine does this species have? 13. Chromatin structure. Histones, organized DNA. condense it, and prepare it for further condensation by non-histone proteins. Non-histone is a general name for other proteins associated with the DNA. Basta pag histones, sila na yung nag-organize ng DNA, and then yung others, non-histone na yung tawag doon. This is a big group with some structural proteins and some that bind only transiently. Ano lang, hindi sila persistent or hindi sila nagsistay. Pag sinasabing transiently, matatanggal din. Non-histone proteins vary widely, even in different cells from the same organism. Most have a net charge and bind by attaching to histones. Ito, mas maganda na mag-refer kayo sa video. Kasi merong mga pictures doon, merong animations doon, and mas maintindihan nyo talaga. So, I will try to find the best na reference, and mas maganda kung panoorin nyo na lang. Both histones and non-histones are involved in physical structure of the chromosome. Histones are abundant, small proteins with a net positive charge. The five main types, H1, H2A, H2B, H3, and H4, by weight, chromosomes have equal amounts of DNA and histones. Histones are highly conserved between species. So, hindi sila pare-parehas ng bilang. So, ayan. Ayun, mag-refer na lang tayo sa video. Chromatin formation involves histones and DNA condensation, so it will fit into the cell, making a 10 nanometer fiber. Chromatin formation has two components, two molecules each of histones associate to form a nucleosome core, and DNA wraps around it 1 to 3 or 4 times for a 7-fold condensation factor. So, ang DNA, magra-wrap siya sa histone natin very tightly. Yun yung nagfo-form ng chromatin. Nucleosome cores are about 11 nanometer in diameter. H1 further condenses the DNA by connecting nucleosomes to create chromatin with a diameter of 30 nanometer for an additional 6-fold condensation. So, ayan. Kaya meron yung term na beads on a string kasi ito yung histones natin. Ito yung histones natin. Ay, bakit ganun? Ulit. Ayan. Ito yung histones natin. Tapos ito yung DNA na nagra-wrap sa histones natin. Parang beads on a string. Mag-gets nyo naman yun. Ayan. So, pag mas tight pa yung pagkakapak nila, magiging ganyan na sila. Ito, sa picture. Beyond the 30 nanometer filament stage, electron microscopy shows 30 to 90 loops of DNA attached to a protein scaffold. SARs, scaffold-associated regions, bind non-histone proteins to form loops that radiate out in spiral fashion. Fully condensed chromosome is 10,000-fold shorter and 400-fold thicker than for DNA alone. Ayan. So, yan na yung 30 nanometer chromatin fiber. Ano na yan? DNA na yan na sobrang na-wrap na sa histones, na-package na. And then, magiging ganyan na. These many different orders of chromatin packing that give rise to the highly condensed metaphase chromosome. So, I think, na lang, itong mga concepts na to, it is better na panoorin na lang natin sa YouTube. So, maghahanap na lang ako ng magandang reference natin. Kasi, this will be better explained by them. Wala akong animations, wala akong pictures na may papakita sa inyo ng maayos para mas ma-visualize nyo. So, for now, the lecture lasted for 30 minutes. Pag may questions kayo, please reach out to me. And then, along with this video, magsasend pa ako ng other references natin. So, yun lang. Thank you.